Focus. What's up, Fish Fam? I was surfing the internet, as people often do, and I came across some questionable claims, and I know what you're thinking at this point. Steven, you come across so many things that aren't true. Haven't you run out of them? Can we really believe that you found another place on the internet where somebody has something that isn't accurate? And yes, as hard as that is to believe, that is what has happened. That is what I am telling you. And I'm asking you to just trust me on this, just this once, as far-fetched as it may seem, while I take you on a very short trip through what I've seen. I was doing some research on filter media, and I was looking at fluidized media because I'm trying to design something that'll go into a big 50-gallon drum and do some self-cleaning, self-turnover stuff, and I happened across a site that was selling colonized K1. I'm going to go ahead and put up an image of it here. How is it different from other media? As the K1 media moves within the filter, it causes the old dead bacteria on the outside to be displaced. This makes space for new, younger, heavier-feeding bacteria to rapidly colonize. Within the wheel is a protected surface which enables colonies of bacteria to naturally follow their life cycle of maturing, dying, and then fueling the latter stage of the nitrification cycle. Now, I don't know how much you guys know about bacteria, but the life cycle of a bacteria is eat, duplicate, no food, hibernate. Food, wake up, eat, duplicate. This happens essentially forever. The scientific consensus has essentially been that bacteria are immortal, that the only way that you kill them is to destroy their cellular walls with something like soap so the lipid bilayers fall apart, or to poison them. And this has been an unchallenged idea of how bacteria work for a really long time. Now recently, there was a challenge to this notion. Somebody did some research on, I believe it was E. coli, where they looked at the actual cellular machinery inside of the bacteria as they replicated. And what they found was, when bacteria replicate, it creates a new clone and it goes floating off over here while this one still sits over here eating. The original bacteria, the parent clone, if you will preferentially keep the damaged cellular machinery that builds up within itself. Historically, we thought that, if there were a lot of damaged amino acids or broken proteins or things, that they would be split with the cells uniformly. And what they found was that the parent holds almost all of it. So each time this parent cell creates a new clone, while they're splitting, it's creating more machinery, and the young one gets a fresh copy, and the parent gets slowly worse and worse and worse and worse. And as this broken machinery builds up, the parent cell becomes less capable of metabolizing. It grows more slowly, it replicates more slowly, it eats more slowly. And over a long enough period of time, the buildup of broken machinery within the cell, in theory, will reach the point at which the cell will no longer function, and it will die in that it'll go into a sort of permanent stasis because it can't metabolize anymore. Now I say in theory because the authors note that the time scale involved in this is long enough that they weren't able to witness it happening during the study. Now, I want to put this into perspective because I feel like that is a very important sentence when you're looking at something like the colonized K1, advertising media. If you start with one E. coli bacteria and you put it in just absolutely perfect conditions, the perfect temperature, the perfect food source, the perfect salinity, all of these things, and you give it the absolute ideal conditions, and you start with one bacteria, at the end of 24 hours you'll have, I think it's something like 16 billion, it might be higher than that. Let me run the numbers. I was a little bit off. E. coli replicate every 20 minutes, okay? Now, that means that in 20 minutes, you start with 1, you have 2. 20 minutes later, you have 4. 20 minutes later, you have 8. 20 minutes later, you have 16. And if you have enough food, it will continue in this fashion. In a 24-hour period of time, this can happen 72 times. And 2 to the power of 72, let's say 71, because the first time doesn't count. That's a big number. I'm not sure even what the name of that is. That doesn't really matter, okay? The important thing is, 24-hour period of time, it has replicated 72 times, in theory. I mean, it's slowed down as it's going through, but 72 replications. If you take the bacteria in an aquarium, the Nitrous Amonas, and you put it in absolutely ideal conditions, and you wait 24 hours, you'll end up with 4. And I don't mean 4 billion, or 4 million, or 4,000, I mean 4. Well, not really. You'll have almost 4. You'll have 2, which are about to split, because they will replicate every 13 hours, not every 20 minutes. Now, if the case of E. coli is that during the course of a study, you cannot have enough replications for this problem to have built up to the point that you would have a cell death for the bacteria, then chances are you or I will die before the Nitrous Amonas in our biofilter does. To explain this claim, you have to know a little bit about the history of Caldinus K1. This media was created for water treatment plants, where you have massive amounts of mucky, gross water flowing through, you know, like a big S for treatment, and not S for treatment, like it's a big S-shaped canal, because that's more condensed, and that's where they do treatment. I know treatment starts with a T. Thanks, Sesame Street. What I mean is, this was developed for this kind of situation, where you have water that is full of other stuff. There's just massive amounts of muck in this water, and it gets all over whatever your media is. And the importance of the self-cleaning function of Caldinus K1 is that it'll scrape the bits of, I mean, it's not sewage anymore at this point, but the bits of biomass off of itself. It'll keep itself clean, so you don't have to take it out and hose it off and lose your bacteria. It will keep the surface area good. It will self-refresh in this environment, and this is the environment in which it was designed to work. It was not designed for aquariums, and they decided to try to sell it to aquarium users later, and that's fine. If you have Caldinus K1 media, it works. Fluidized media beds are a good idea. You don't have dead spots in your filter that way. But they don't work the way that they claim they do. They work in the way that if you've got a sump, you don't need a pre-filter sponge, because they're going to clean the crap off of themselves. That's what it is. If you have a pre-filter sponge, then there's no reason to go with Caldinus, because your water's going to be clean enough that they're not going to do anything, because their claim about bacteria is faulty. And even if we pretend that's not the case, and it is, but even if we pretend it's not, the bacteria that we're dealing with are at the largest like 6 micrometers wide, which means that these things are so small, it doesn't matter how often you bump, you're not going to actually interact with the bacteria. That's the entire way that it can function, is they're not going to bump the bacteria. And you can't just shake bacteria off of something. If you have a stick and you stick it in poo and you shake it around a whole lot, the bacteria is still there. It's not going to come off just because you smack it on the wall. It's not safe to put in your mouth just because you shook it in something. And I want to say, it's my opinion of this, because this company is flatly lying to you, and this isn't just them, a lot of other places have repeated this claim. I remember a couple years ago Joey did something on Caldinus K1 Media, and he said exactly the same thing on his channel, and it didn't occur to me that it was garbage at the time, because I wasn't busy nitpicking everything in the universe that has to do with fish, but I am now, for you. That's the sacrifice I'm willing to make for my viewers. Now, it's something that I just didn't think about at the time, because it was just background noise going on. I don't know what video it is. I'm not going to put a clip in here because I'm not criticizing him right now. I'm not criticizing any of the other YouTube channels that have repeated this, because these people, it's not their job to know better. It's the job of the people that make this stuff to be honest. So, you know, if you like fluidized media, there are a lot of options. You can do fluidized sand beds. You can do styrofoam pellets. You know, the things they put in beanbag chairs, the little styrofoam beads. I mean, we've probably all had beanbag chairs as children that we tore a hole in, and then the dog came and ate it, and you ended up with big white poop all over the place. That's not just me, right? That can't just be me. Anyways, I'm sure that you guys know what I'm talking about in that. That stuff works really well if you don't have sludge in it. You know, if you just dump the fish tank water directly into it, you're going to have bits of fish poop and other stuff stick to it. It's going to stop working. In that situation, the coldness stuff works. But if you have that situation, I would strongly suggest that you buy the Chinese knockoff brand, because any company that's going to lie to you to make a sale doesn't deserve your money. Until next time, everybody take it easy. If you like this video, don't forget to subscribe. Go ahead and hit that thumbs up button.